Hello and welcome, dear students. Let's get back to our discussions regarding genetics. And today I would like to discuss a very interesting aspect with respect to the changes in the DNA, and that is called as mutations. So, in this lecture today, I would like to just introduce you to the concept of mutation, how this mechanism evolved in the minds of the scientists, what actually is mutation at the level of DNA, and what are its basic types. Of course, as the syllabus goes on, we will be dealing with great detail about the various types and other perspectives regarding this very important phenomenon which governs the process of evolution. So, come, let's see what mutation is all about. So, if you see the background of this whole idea of mutation, it came from Darwin, Lamarck, Mendel, etc. Darwin's origin of species and the Darwinism concept. If you go back in the history, in 1859, Darwin published his very important work, and he compiled it in a book called as On the Origin of Species by Means of Natural Selection. All organisms, he says, have been descended with a modification from a common ancestor. That is, everybody's ancestor was one. Means when you talk about the origin of life itself, somewhere there has to be one single unit of life which gave rise to multiple forms of life. So he introduced the theory that populations, they evolve over the course of generations through a process of natural selection. Means in general we all know the survival of the fittest theory. In view of this Darwinism theory, the hereditary variation which we observe, that arises by fluctuation amongst the progenies, and evolutionary change, it accumulates automatically in over the time, he says, and fluctuations, they just go in the favorable direction, and the environment, it plays a very important role. So what we are discussing here when we are talking about Darwinism or Lamarckianism or Mendelism, we are talking about evolution of species. Now what is evolution? In the genetic language, we are talking about change in the trait. So various perspectives of this change, okay, today we know that this change is majorly due to the process of mutation. So historically, if you just see, there are various scientists who put forth various theories of mutation, but Hugo de Vries, his theory holds much more what you can say closer to what we believe in today. According to these scientists, or Darwin, you can say, sudden appearance of a new hereditary character in the progenies, means when the species is growing, multiplying, next generation is being formed, you see sudden new characters. So he referred to that as a sport or a mutation. According to Sinott and others, in a very broad sense, they explained it in macro mutation and micro mutation. They said that any heritable change in the genotype, not the genes, the genotype, means the expressed character, is called as the macro mutation, and in a narrow sense, mutation is the change in the gene, they say, or change of the gene. So that was the concept of Sinott. According to Batson, here I am not giving you the definitions, try to understand. We are just trying to understand various perspectives of scientists. Today, what is a definite statement for mutation? I will explain it in the next slide. So here you just want to know what were the ideas which existed in the minds of the scientists who were philosophizing or professing or studying the phenomena of mutation. Also try to understand that this study or this process of thought doesn't have any instrumentation or laboratory. It is purely based upon observations of various species. Just like Darwin, he went to Galapagos Islands, and there he saw various, he made drawings of beaks of finches and all these things. So it was pure observation and a philosophy of the highest order of thought. So we are having various scientists. So as I said, according to Batson, Mutation is a discontinuous variation, he said. Means it is not in continuum. You can see that the variation, it is split, and it may be there in one generation, it may not be there, and it takes a huge form of variation. So he said mutation is a discontinuous variation. According to De Vries, now this is somewhat what we can say correlated to what we believe in today. Mutations are sudden and drastic heritable changes, he said. This we believe it today to be true. Again, at this point, with respect to De Vries theory, I would like to tell you that the study of evolution is a continuous process. It never ended. It has not ended till the time I am giving you the lectures. The thoughts and philosophies and the discussions are going on and on. So according to De Vries, he said mutations are sudden and drastic heritable changes, not traceable or credited to segregation or recombination. Okay, these changes, these are sudden and heritable, but they are not attributed to the crossing over of the chromosomes as we see in mitosis and meiosis. You see, all these theories are with respect to the eukaryotic systems only, because till the time we understand what the Mendelian genetics is, classical genetics is, we come to a point of time where we talk about molecular genetics, and there the picture becomes more clearer. When we can play with the DNA today, picture is very clear, but we are taking a time in history and trying to understand the psychology and the philosophies of those people who were thinking on mutation in those times. So even at that time, De Vries, a Dutch botanist, he said what is true today: mutations are sudden and drastic heritable changes, not traceable or credited to segregation or recombination. We know these sudden changes are today due to genetic modifications of the bases, and maybe with respect to some environmental change also. Stebbins, he described mutation as a discontinuous chromosomal change with genetic effect. He further states that chromosomal change refers to chemical change in a small part of a chromosome, as well as the alteration of the physical structure of the chromosome. So this was, again, if you see now today the chemical and the physical mutagenic agents, maybe their part of that is true. Amato and Otto in 1956, they defined mutation as a change in the hereditary constitution of a given species. Now, a change in the hereditary constitution, a complete makeup of the genotype and the phenotype of a given species, whatever the change you see, that is mutation. Of course, all these, when you combine all these theories and philosophies, you today define mutation, which we understand very perfectly, very clearly, is a process which affects protein synthesis. Whether it is a eukaryotic protein synthesis or a prokaryotic protein synthesis, mutation definitely alters the protein as a final product. So we are somewhere talking about translation, we are somewhere talking about transcription when the changes are introduced into the gene. So in biology, now this is almost a exhaustive, comprehensive, complete definition of mutation. In biology, a mutation is defined as an alteration in the nucleotide sequence of the genome, means the DNA of genome. Whose DNA? It can be any organism, virus, microorganism, humans, plants, across the species, across the life systems, wherever DNA is there. A process where alteration in the nucleotide sequence of the genome of an organism, virus, or extra chromosomal DNA, which results from errors during DNA replication. That is, errors during the DNA or the viral RNA, you can say, or viral replication, when it takes place during maybe you can say in bacteria, that is binary fission. In eukaryotic system, mitosis, meiosis, whenever the DNA is trying to replicate, make more copies of itself, errors in the DNA replication, they occur. If you recall the DNA replication, one base in 1000 base is wrongly added. The DNA has a perfect methylation and editing function, due to which it does not allow the wrongly incorporated bases to remain. But still it remains, and that is what we call it as a mutation. Such as the pyrimidines or purines, they can be misplaced, or pyrimidine dimers can be introduced. We will see all this in the types. So which can result in insertion of a base, deletion of a base, insertion of a segment, deletion of a segment of a DNA due to mobile genetic elements like transposon. They are also one of the reasons for mutation. So if you just simplify it, mutation is a sudden change due to the alteration of a nucleotide sequence in the DNA of any living system, due to which there is a change in the altered sequence of the DNA. So there is a change in the sequence of the mRNA, and there is a change in the sequence of the protein. So that is what we define mutation as. Now what does this mutation means? What it actually have a significance with respect to observable phenotype and the non-observable systems like the metabolism. The progenies which are there, if you are talking about a bacterial cell, it's next progeny, or if we humans, then the offspring, our offspring, they resemble their parents, okay, in one or several respects. Yet you can see, except for the bacterial system, in multicellular system there are differences. Even in the bacteria there can be differences. You may have, if you see the stationary or the death phase cells as compared to the log phase cells, those two cells in the same colony, they may be different. So the offspring, they may resemble their parents in one or several respects, but there exist differences in between these two. These differences, whether large or small, are called as variations. Now variations, this may be induced by the environment or they can be hereditary. When you talk about induced variation, it can be environmental or it can be hereditary. See, hereditary variations are permanent. Okay, those caused by the environment, they are not permanent. Whatever the variations they appear due to change in the hereditary mechanism, they are permanent. Not only permanent, they are transferable to the next generation again. Similarly, if you see the variations caused by environment, they won't be transferred to the next generation. That is the difference. So a sudden appearance of a marked heritable variation in the nature of any organism is called as mutation, and that organism or that living system will be called as a mutant. A mutant or an individual or a cell, it can be a mutant individual or a mutant single cell in which there is a changed phenotype. That changed phenotype is definitely attributed to a changed gene or a mutated gene, and that gene, when it is mutated, will result into a altered protein, which may be enzyme, which may be a hormone, which may be a very important chemical responsible for the health, and the health may be altered. So the mechanism of mutation is what I just said. This heritable change is in the basically in the DNA sequence of the organism. This mutant organism with a changed phenotype, what is the opposite of a changed phenotype is a wild phenotype, means an organism which is not changed, it is as its original state, is called as a wild type organism. These are the genetic jargons, the genetic what you can say terminologies which you should remember. And this organism which is called as a mutant has a recognizable changed phenotype, and this phenotype is commonly observed in nature. The change in the DNA sequence which is there, that is through this mRNA. Okay, once the DNA is formed, when there is a changed altered DNA, mRNA is altered. mRNA has a coding sequences. Change in the DNA sequence conferred through mRNA, that is due to transcription. Through transcription leads to an altered amino acid sequence in the protein or during the translation process. Now as the protein is altered, they carry out majority of cellular functions. Proteins, they carry out majority of cellular functions. As they are altered, a change in the phenotype is observed. It can be a change in the phenotype, and if it is non-observable, it can be change in the genotype leading to diseases. So this is the mechanism of mutation. To sum it up, mechanism of mutation is with respect to transcription and translation. A changed DNA will result into a changed mRNA, will result into an altered protein. This is the basic mechanism of mutation. Now if you see the classification of mutation, it will be naturally on the very basic level classified whether it is a chromosomal DNA or a naked DNA. Naked DNA will be found in prokaryotes, and if there is a alteration in the naked DNA that will be found in prokaryotes. So chromosomal mutations, if you see, these are, now this is I suppose this is a slide where eukaryotes and prokaryotes have been misplaced. So don't see, this eukaryote should have been here and prokaryote should have been here. So this I will refer to as eukaryotes. So just make a correction while observing here on the screen. So mutations or changes in the gene or chromosome, that is, chromosome is a concept which is mostly associated with eukaryotes. So chromosome is eukaryotic in nature. So change in the number of chromosome or change in the structure of chromosome is one type. Chromosomal mutations they are called as. Change in the number of chromosomes like diploid, polyploid, aneuploid, these are change in the number. We have 224 sets of chromosomes. Two chromosomes you have, one set, and we say there are 24 chromosomes. So there are alleles. This concept is not there in bacteria or prokaryotes. So we call there as gene mutations, simple gene mutations in bacteria, chromosomal mutations in eukaryotes. So when you talk about a gene mutation, the replacement of one nucleotide by another, addition, deletion, we will be seeing this in great detail in the next lecture, and in that next lecture I will be rectifying this slide. So here just you make the eukaryotes in your mind at this point and prokaryotes shift the places. So chromosomal mutations are in eukaryotes, change in the number of chromosomes, and gene mutations are in prokaryotes. So this is the basic classification of a mutation. But when you talk about types, what happens in these basic types, whether it is eukaryotic or prokaryotic, what happens there? A mutation usually is due to altered nucleotide sequence or a nucleotide base. How many bases are there? Adenine, guanine, cytosine and thymine. 4 bases are there. So we have 2 types of gene alterations resulting due to modification of the bases. Okay. What happens if a AT, which is a normal Watson and Crick pairing, adenine binding with thymine and guanine binding with cytosine, this is the normal Watson and Crick pairing. If this pairing is altered, AT instead of AT it becomes AC. Now the question may arise, why it will become? There are reasons. There are inborn errors in the metabolism. During metabolism, many chemical changes, chemical shifts, and many alteration in the bases, modification of the bases automatically takes place, and it is huge. But the cell has a very efficient repair system. We have editing function of the DNA polymerase itself, due to which these errors are minimized to a great extent. We are talking about that rare event when this alteration sustains, it remains in the whole process. So if adenine is being, that is AT, A is converted to T, T is to K, means we are talking about transition and transversion, means purines and pyrimidines. If purine is replaced by another purine and a pyrimidine is replaced by another pyrimidine, we call it as transition. And in transversions, a purine is replaced by a pyrimidine and a pyrimidine is replaced by a purine. I will be giving these examples in the type, but here I just want to explain in broad the various types of mutations. So mutation, it can be transition or transversion depending upon which base is altered. Then on that mechanism, you have base substitution or point mutation, which can be transition or transversion. You can have frame shift mutation where the whole reading frame is shifted. In frame shift mutations, you have addition and deletion type of mutations, that is addition and deletion. Whereas in base pair substitution or point mutations, you have silent mutation, missense mutation and nonsense mutation. In missense, there is acceptable, partially acceptable and unacceptable type of missense mutations. This is all with respect to the protein synthesis which is going on. In my next lecture, I will be detailing about this. See, this is just to give you a basic concept of mutation today. Now the question is why this transition and transversions may occur. I will be explaining this as I said, but here you can see these are the common forms of the 4 bases. You have thymine, you have see the laser marker, follow the laser pointer. Thymine, guanine, cytosine and adenine, these are normal common forms. Okay, these are the common forms. Now what happens if a proton is shifted? You have a rare form of thymine. This H from the amine shifts to carboxyl, it becomes OH. Now these are the inborn errors in metabolism or inborn changes, radical change, free radicals. They also introduce changes. So this is a rare form. We call it as a immunoform or the enol form. So again you can see guanine, there is a proton shift. In cytosine there is a shift. These shifts can give rise to rare forms of these bases. Now if you see the common base pair arrangement, adenine, thymine and guanine, cytosine, this is with 2 hydrogen bonds, this is with 3 hydrogen bonds, this is a normal Watson and Crick pairing, the common forms. But these rare forms, if they come into play in the nucleotide, if there are rare forms, then what happens? Unusual base pairing like cytosine pairing with adenine and guanine pairing with thymine may take place. These are the reasons why mutations occur. This is the basic fundamental reason. What it can be? The alteration of the bases. And alteration of the bases may be due to various reasons. Physical reason, chemical reason, that will be explained further. So the next concept is mutation rates. What is the rate? What is the rate of mutation going on in nature? If mutations are so common, then we would have seen a great change in the phenotype and the genotype of all the species which we are observing, but it is not so. In genetics, the mutation rate is defined as the frequency of new mutation in a single gene or the organism over time. Means for how many generations? If I want to give you a small example here, if we were believed to be evolving from chimpanzees or monkeys, then we don't have a tail today. It took some million years for us to lose the tail. Why? Because we developed our hands and we started using our hands. What is the use of the tail? If you just observe the animals with tail, they swab the flies, they protect their wounds, and somehow they use it for their what you can say, defense mechanism, protecting the wounds, swatting the flies, etc. Okay, so even up to some extent they keep their anal covered, their anal covering it is meant to be. But as we grew up, as we started developing our forelimbs and hind limbs, we didn't find any use of the tail. So maybe in 1 million years we lost the function of the tail. The proof is the tailbone which we have in our vertebral column. There is a tailbone. Okay, so the point in discussion here is that in evolution, during evolution, there is a certain frequency which mutation results into a loss of a trait or a gain in a trait. Mutation rates are not constant and they are not limited to any one single type of mutation like one trait. It can occur throughout the character or throughout the trait. Therefore, there are many different types of mutations. Mutation rates are given for specific classes of mutation. Mutation rate of organism, it evolves characteristically and it is strongly influenced by the genes and the genetic makeup of each organism. In addition to the influence of the environment, you should take into consideration mutation rate. It varies. Means what is mutation rate? In which generation you are going to see a permanent change. If I try to go back to my original ancestor who had a tail, then I will see that it had hundreds and thousands of generations. So after this generation you are having a permanent change, no tail. So that is a permanent change which a generation acquires, that is the rate. So it will be one generation in or one particular cell. If you are talking about microorganisms, one cell in 10 raised to thousands and thousands of cells. So how much that is, we have found out the frequency. I will give you it in the next slide. So try to understand mutation rate is defined as the frequency of a new mutation in a single gene or a single organism over the time. And it is influenced by hereditary changes and environmental changes. There are spontaneous mutations, there are induced mutations that we will discuss in the future. So when to give an example, when the mutation rate in human increases, you can say certain health risks are there, like for example cancer. Cancer is a result of gene alteration and other hereditary diseases which are linked to DNA. As the mutation rate is very fast, you can see those diseases in that generation itself. but it is not really necessary that you will find that disease in each and every generation of his progeny. Okay, so point here is there is a certain mutation rate at which this mutation goes on in the DNA and in the generations. Having the knowledge of mutation rate is very important to understand the future of many hereditary diseases. Okay, and that is what we are using microorganisms, because microorganisms, they have a rapid multiplication rate and they are our ancestors somewhere. You can say that that is origin of species. If you see the tree of life, somewhere all the organisms, they are related to one another. So here understanding the microbes gives a great insight to the evolutionary mechanisms of DNA, and we can formulate newer drugs, newer medicines for us to cure. The rate of these types of mutations, they are further divided into a mutation spectrum, a concept called as mutation spectrum. It's not in your syllabus, but for those interested, you can study this. A mutation spectrum of an organism is the rate at which different types of mutations occur at different sites in the genome. Means if you call them as hotspots. So what are the points where mutation is greater as compared to other site? This is called as a mutation spectrum, and that is giving great insight for drug designing, for to find out novel medicines for the novel diseases which are coming in every generation. So this is mutation rate. Now let us see what are the different mutation rates in bacteria, viruses, etc. That is called as the rate of mutation. Means one sustainable permanent change which is observed in the generation. Mutation rates, they vary greatly across species. In humans, the mutation rate is about randomly, means de novo, or newly formed. De novo means newly formed mutations are about 50 to 90 per genome per generation. Per genome per generation 50 to 90 mutations are there, and in each human he can show 50 to 90 base per generation, new addition, deletion, substitution, whatever it may be, which was not there in his father's genes, and the son may show which was not there in his grandson's genes, so on and so forth. Means every generation, that is very less actually, but still 50 to 90 per genome per generation changes are there. Some sequences of the DNA may be more susceptible to mutation, like for example, if you see, this is a very interesting aspect, all of you should understand. In human, mutation rate is very high in the male germline as compared to the female germline. Means the female egg cells, they are permanently structured, they don't undergo much mutation as compared to the human sperms. So human sperm, just because it lacks methylation, one thing, it lacks methylation, and second thing is that the human sperm is very prone to mutation. So whatever the genetic birth defects, etc., these are being found in most of the species. The reason is males. It is not the females who are contributing more to the mutation, because the egg cells, they are not that prone to mutation as compared to the sperms. So what I mean to say here is, some sequences of DNA, they are more susceptible as compared to the other sequences of the DNA. In unicellular eukaryotes and bacteria, unicellular eukaryotes, that is the small eukaryotes like the blue-green algae, etc., roughly one cell or one gene per generation. In 1000 cells, the mutation is permanent. Means that is 10 raised to minus 3. One cell in 1000 cells. So if you express it, that is 10 raised to minus 3, or 0.003 is the rate of mutation. Similarly, if you see bacteria, the bacteria, they exhibit mutation rates from 10 raised to minus 6 to 10 raised to minus 12. Means minimum one cell in 100,000 cells. Okay, not only 100,000, 1,000 cells. Okay, one cell in 1,000 cell of bacteria, minimum to 10 raised to the 12 power 12. That many generations, only one bacteria, it may undergo a permanent change. So it is very low. Rate of permanent change is very low unless there is a induced mutation. If environmentally induced mutation is there, this rate of mutation can be enhanced, but that is not permanent. It will not be transferred to next generation. Immediately next generation will repair it. In microorganisms, all the genes that are involving the phenomenon of transposition, transposons, they result into mutation. So wherever there is a transposition phenomenon, that becomes a hotspot gene, and there is a mutation. DNA viruses, they have mutation. DNA viruses, they have mutation 10 raised to -6 to -8, and RNA viruses, they have a mutation between -3, 10 raised to -3 to 10 raised to -5. Means one cell in 1000 cells to one cell in some 100,000 cells, it can undergo a permanent change. This is called as the rate of mutation. Finally, let's talk about the role of mutation in evolution. It will not be exaggeration. It is a widely accepted statement that mutations are raw materials of evolution. Understand this. Wherever there is evolution, it has come there as a product of mutation. Mutation is the ultimate source of all genetic variation. Okay, providing that mutation is the reason, I can say only reason for mutation, which provides the raw material for all the evolutionary forces where the natural selection can act. Mutation may or may not produce a detectable change. Okay, means a phenotype may be observable, it may not. Like diseases, it takes a long time for the onset, and till the time the change which is there, it is not observable. Third point is that in evolution, if you say mutation, it plays a part in both normal and abnormal biological processes. Means on one side there can be evolution like cancer, okay, cancer which is abnormal, whereas very normal like development of the immune system. Suddenly you can have a very strong immune system. There is corona going on, a pandemic, and suddenly you find yourself very prone or resist. Some people may be resistant to it, so that is they are having a good immune system. So that is also a mutation. Okay, mutation can result into many different type and change in the sequences. Mutations can affect the phenotype, may not affect the phenotype. It depends totally upon the evolutionary mechanism of that gene of that species in time. Due to the damaging effects of mutation, the organisms like the prokaryotic organisms, they have developed a mechanism called as repair. So we will be seeing what is DNA repair. Evolution has changed the horrible result of death of the microorganisms into evolution, mutation, repair, DNA repair, and they have survived all the harsh conditions on this earth. Okay, finally, with respect to evolution, we can say the duplications of DNA, which are major source of raw material for new genes, and as tens and thousands and hundreds of genes are duplicated in animal genomes every million years, this one permanent change does result into a modified phenotype over the period of time. So mutations, they are the raw materials of evolution. What effect these mutations it has on a phenotype? As I said, it can be hazardous, it can be leading to cancer, or it can be lead to a increased immune system also. So based on the survival of the individual, either whether it is a plant or a microorganism or a human, the mutations can be categorized into 5 types on the basis of the effect which it has on the survival of a phenotype or the survival of an individual. First is lethal mutation. When the mutation causes the death, simple cancer, etc., even in microorganism, the death phase which comes, all the DNA it is altered and there is great mutation going on and the cell dies. So when mutation causes death of an individual undergoing mutation, it is called as lethal mutation. Sublethal mutation is it causes death of the maximum fraction of the population, but not all. Some they become mutated and somehow they survive, branching off to a newer life form. So that is sublethal mutation. Subvital mutation, these mutations which kill less than 90% individuals, they are subvital. And vital mutation is the mutation which does not affect the survival at all. There is a mutation but it does not affect his survival. Okay, like a somewhat temporarily increased immune system for this pandemic. If there is a temporarily induced immune system for this pandemic, then that is vital. But if he gets a immune system which is super for all the diseases, then that is super vital mutation, the kind of mutation which enhances the survival of the organism. So these are the effects of the mutation based upon the survival of an individual. Finally, these mutations, one philosophy which you can students learn from the mutation is that, for off the track, I would like to give a very nice quotation. If we did not have any genetic mutations, we would not have us ourselves. We are a product of mutation. So we are what is mutation? Mutation is error. So you need error to open the door to to the adjacent possible event which is there, the near possible event. If you want that to happen, and for a change, for survival, for sustaining, there has to be mutations. So don't worry the errors. That is the message of this slide. Don't worry the mistakes which happen in your life. Maybe they are opening to newer vistas, newer vital and super vital possibilities in your life. So that is a quote by Steven Johnson. In our next lecture, we will see the mutation types in detail, the different types of mutation, and how they are categorized with examples will be the point of my next discussion. Till that time, I hope you understood and enjoyed this lecture as a part of your curriculum. Till then, goodbye, study hard, and make these concepts clear in your mind. Goodbye.